And this is associated to my experience with Hemiptera bugs, in particular planthoppers and leafhoppers, that are probably the animals, or the insects in particular, that use more this kind of communication. So during this talk, I will talk about biotremology, what is biotremology, why is it important, and how it's possible to do applications. Because unlike chemical ecology, that has a long story nowadays, it's more than 30, 40 years that people are using chemical ecology approaches to control pests in different agricultural systems. There is unfortunately nothing like that in the field of the biotremology. And we think that now the time has come to open this new opportunity, this new path, to offer to the farmers and to, of course, the society, new tools, new devices to help the control and the management of the pests. So I start from, of course, geography, and the place where I live is Italy, and in particular this red point that is a Trentino. That is a region, as you can see, in the middle of the mountains. And here around, I have, if I show you my window, we have peaks of 2,000, 3,000 meters. That is barely, I would say, 6,000 to 8,000 feet. So you can imagine a region made of many valleys like this in the picture. And this is the place where I work. And as you can see, there are villages, rather small, between 1,000 to 10,000 inhabitants, but all around there are orchards and vineyards. So it's very important in a situation like this to have a management that is respectful of the people. You can imagine that if you open your window and you find somebody spraying some pesticides, insecticides, you are not happy. However, this is one of the most important, maybe the most important economical factor of the region, together with the tourism, of course, because you can imagine that people come for tourism, for skiing, or for climbing. So it's very important, the quality of the environment here. And for this reason, we need to manage in a sustainable way, in a way that's respectful of the environment. So one point of honor for us is that 100% of the surface covered by vineyards, is about 10,000 hectares, is covered with the mating disruption technique with the pheromones. So our region is totally managed with the pheromones to control grapevine moths. And this is something that is not happened in one day. It took about 15 years to get to this level. And in 15 years, we covered the full surface, and we clearly drastically reduced the number of insecticide treatments. Basically, when we reached the 100% of surface, the insecticide were, I can say, totally eliminated by our management strategy. And this is a very good point, of course. These are some techniques of dispensers or spray, the type puffer sprays that are in less number, but they cover a higher surface. So basically we were happy. But one day something happened because alien invasion. Well, aliens is one of the most important threats that our society is facing in the last years. And among these, we have three main characters, in Italy at least, but at least one is shared in the USA, that is Daliomorphalis, the brown-banded stingbug. And then we have other two, that is the Drosophila suzuki, that's also shared with the USA. They both come from Near East, Far East. And another one is a leafhopper, Scapoideus titanus, who is from North America in this case. In North America is not relevant at all, but here is a bad vector of a phytoplasma disease that is called flavescens doré of the grapevine. And this is a quarantine disease. So basically, when these guys came to Italy, to Europe, we had to face new problems. We thought that with the pheromones we solve problems, no insecticides, but indeed this is not the case. So I want just to give some introduction about the signals and why are they important. And I start with a very, very basic problem about life meaning. And basically all animals and insects in particular have two crucial aims that are to survive, so to reproduce. Of course, this means food and sex. And what we do here is to study the sexual part, because from these factors depends selection and then evolution in the long term. But what is even more important is the searching that they use, the searching behavior that they use to find a suitable partner, but also edible food. So the mating behavior is one crucial part of this pattern, and the knowledge of the mating behavior is very important to establish any strategy, especially when you're facing new elements like alien species that basically most of the time we don't know anything about. And if we don't know the mating behavior, of course, also the edible food is very important, and the phenology, and many other aspects. But in our case, we focus our activity on the mating behavior. And what are the kind of information we find in the signals that these animals use during mating behavior? Basically, it's information about identity and also quality and position, so location in the space. And from these points derives three important factors that are the co-specificity, partner choosiness, and also it's very important the active space and how to maximize the range of your signals. I'm going fast, but unfortunately we have only a few minutes. But if you have at the end any question, I'm very happy to respond. I know that I'm going fast, but let's try to do the best. So altogether they give the selective pressure. The selective pressure given by the environment, of course, by the host plants, and also by the conditions that a certain area can provide. And altogether, we have sexual selection and finally environmental adaptation. Now we have to think that these new alien species, they have their pattern of behaviors, signals, and they are sort of thrown in a new place, and basically we can't predict if they will have success, we can't predict if they will be dangerous, if they survive. And this is something, this is normally a problem, because when I go to my administrator or people of the politicians that should decide if I have to invest in a new alien species that just arrived, as long as we don't see real damage, they don't give you a dollar, because they say, oh, this is not a problem. How can you predict that it will be a problem, and so on. And this is something that is happening very often here in USA, because last week was in Pennsylvania, and they told me that the problem with the spotted lanternfly has become very important, because in the beginning nothing was done. That was they told me, and I say, yeah, we know the problem. So basically, the actual signal of an animal and the possibility of the signal to be successful depends on all these factors, and the success of a species depends from many factors that must be known. So a good signal must be charming, of course, having a wide range, but in compromise with other costs. Normally, just to say in a few words, it's energy consumption, but also predation risk that is associated to it. So if we talk about the biotremology and how biotremology can stay in this pattern, first of all, I want to define a biotremology. There was a very nice paper of Hill and Vessel in 2016 that was a primer of this on current biology of this new discipline. Biotremology is a new discipline, and it's different from bioacoustics. The figure is very clear here that when you have any animal that is sending a message, there is an airborne fraction, but also a substrate component. And according to the animal, in the case, for instance, of the plant toppers, the substrate borne part is much more important. We did some studies, and we found, for instance, that for some leafhoppers, there is a range of 6 to 7 centimeters when the transmission can be even done from a surface to another. In that case, it was a couple of leaves. And so there was an higher transmission. But on the other hand, there were sort of 20, 30 centimeters of active space on the surface. So we should evaluate both of them when approaching a new species, but we know that in the case of this kind of species, plant hoppers, leaf hoppers, also stinkbugs in many cases, we have a much more important part in the vibrational component. And yeah, here we have Peggy Hill, that is probably the mother of this science. She's a very nice person. She's from USA. And I think that the biotremology, good part exists because of her. Not only, but she did a lot about that. So what's important to distinguish biotremology from bioacoustics? But of course, because I already told this, because the signal that is traveling to reach a receiver use a different matter. So in the case of these are a couple of Aphrodis, they are leafhoppers that are studied because they are also a problem in agriculture. So we have a sender that has some issue to solve about the production of the sound of the signal, in this case, the substrate borne signal. And it's very important also to associate the necessary intensity to enlarge the active space, and to transmit the transmission properties of the plants, of course, are also very important, because not any surface is the same. There are problems with attenuation, filtering, degradation, also considering the frequency pattern that can change a lot according to the substrate. While the receiver needs, of course, receptor mechanisms that are adapted to this kind of signal reception. And then there are problems connected with the threshold of intensity, the frequency response, and everything is connected with the central nervous system, of course. So this is the basic picture, and we can transport the same concept to other sensory modalities, but of course, there are peculiarities for each of them. Sensory organs. Well, there are many sensory organs that were studied, and probably they will find others in the future, because very few species have been studied indeed. So there are slit sense organs in spiders, for instance, but the most common in many insects is the subgenual organs that are in the legs. Normally in 2, 4, sometimes also 6 legs, depends on the species. And these are the organs that are more studied for this kind in the biotremology field. Now, I want to show some videos, hopefully they will work well, about example of signal transmission. So these are lice wings, is a chrysopa, and we recorded here as example of drumming and percussion. So probably the audio is not working. I try to unplug it here and see if it works again. Let's see if it works, but no, it doesn't work. But what is important in this case is to see here that the abdomen, in the case of the timbal buckling, that is an organ that is inside the thorax and abdomen. While drumming and percussion involve the contact between body and the surface, and tremulation is a trembling behavior that allows the transmission of vibrations from the body to the surface. Timbal buckling is a more specific, more specialized organ, and is inside the body of the insect. And then there is another way, that is a stridulation, that is just the scratching, the scraping between two. I can try to see at least. You see, this is a behavior typical of ants, and here you can see that the ant is scraping the abdomen with the thorax. There is a plectrum and the pars stridens, that are the two parts that are scratching each other, and they make this sound. I'm sorry it's not working, the audio, but we tried before, it was working, now it was not working. So, is vibrational communication something awkward, something unusual? No. If we consider, this is a phenology tree, rather recent, from 2012, so even more recent, of course. But so consider that airborne sound communication is very, very frequent in grasshoppers, and then we found other few orders or groups that use commonly airborne sounds. But what happens if we consider vibrational communication? You see that blue and red, common and present, are spread everywhere. So this means that the vibrational communication is a rule. It's not something that it's unusual. And so that's a good reason to study it more in depth. And now that we have technologies to do this, it's very worthy to do it. There are, more than 200,000 species that we know that can do that. Another important aspect when we talk about vibrational communication then is the active space network, and also the interaction of different animals in the same environment. Well, we know that an active space can be defined like the surface in the case of biotremology, of course, where a signal is received by a receiver that can interpret the signal in the proper way. This means that basically, in the case of many insects, the active space corresponds with one leaf or a branch. Very few cases, a full plant or another surface. I'm focusing in this moment on host plant feeders, of course, because then the point for me is to arrive to talk about how to manage vibrational communication for pest control. So there are many other cases about biotremology, but in this moment I'm going to focus more on this kind of animals. And of course, you can find an environment intended receivers. Of course, a male normally calls for a female, not for rival, not for predators or parasitoids. So, however, you have to imagine that there is an eavesdropping network all around where anybody can take the cue and reach the sender before, for instance, this guy has opportunity to mate. So it's a risk in the environment to send signals, and that's the reason for which many of these animals use only a short part of the day to communicate. Many animals are nocturnal, many others, like Scaphoideus titanus, that is our model species, we talk about it later, only 3-4 hours during the twilight, so when the sun is going down. So you can understand this is a strategy to avoid to be absorbed by other animals. And what are the information that are given by substrate-borne signals? Well, first of all is who and where, because the signals are very specific. In most cases are species specific. So it's a signal that involves a code, and a male address it to the female of the same species. Of course, this signal also gives information about position, because these animals can track back the signal and find the source. This is very important when you think about trapping system. We are developing now a trap for the brown stink bug that is based on this principle. So it's very important to know what kind of communication characterize your target species, because according to the behavior, you can use a different strategy of crop protection. In the case of Scaphoideus, for instance, and I will tell you why in a short, we chose a mating disruption approach, while we discarded a trapping system. Exactly the opposite in the case of brown stink bug, because the behavior was different, the signals were different, and it was not possible at all to do any kind of mating disruption, but the males are very attracted by the females. And in this case, you can develop a very nice trapping system. So I just give you some example of other behaviors, because there are tons of behaviors that are driven by vibration and communication. And in particular, prey host detection is very important. There are predators that use incidental vibrations to locate the prey, but also parasitoids that are so not generic predators, but they are very specialized as a parasitoid to find a specific host. So they need to interpret in a very, I would say, sophisticated way the signal that they detect in the environment, because they need to reach the X of the host and not any kind of egg. And of course, there are also generalist predators that, however, can detect a specific signal. So there are predators that are also specialized in recognizing specific prey. Other behaviors. Well, recently a paper about the synchronization of hatching. So if one egg starts hatching, and if you belong to a species where it's possible to have cannibalism, of course it's your own interest to hatch very quickly. And the same happens when there is a nice paper of Heron Barkentin about hatching of frogs, tadpole, that they start to hatch when a snake tries to bite one of them. All the others start hatching. There are nice videos on the web about that. And of course, also predator avoidances. So you have one guy that starts signaling the others the presence of a predator, and then they just leave the plant falling down. I want to also say something about a peculiarity of the, this is also common to airborne sound probably, but in the case of a substrate-borne sound, where the environment plays a very important role, you can find a lot of dialects. So there are, this species is Nesara viridula, it's a stink bug, the green stink bug, is a worldwide spread, but if you, genetically they studied, it's the same species, but the signal is different according to the region. So I understand that it's very important also to characterize the signal of the species in your region, not just getting information from Japan, hoping that this works also in USA or in Italy. We have to work and maybe make comparisons, of course, and see if the language is the same. So making disruption, what does it mean, making disruption? I want just to give some parallelism between the mating disruption with pheromone and mating disruption with vibrations. That is the new discipline, the new approach. So if you see the story of chemical ecology and pheromone disruption, you see that everything started a long time ago, and you had the first registration of a pheromone for mating disruption in 1978. So you see that the story is long, but if you go and then you have the last few years, many more registrations. But if you go to the vibration and disruption and see what has been done in the last century, for instance, well, the first the first proof of the existence of vibrational communication in 1949. This is 50 years or more late after, 70 years after the first discovery of pheromones. But, and also the demonstration, that 1949 was a hypothesis, but the first real demonstration 1974. So I understand that that's a very big difference between the two things. And what about papers? It's easy to see here that the number of papers on chemical ecology and on vibrational communication or biotremology, it's very, very distant. There is a big gap. So our discipline is a newborn, and there are very few scientists now doing that. So that would be a good opportunity to do something new for sure. Yeah, the paper of Hill and Bessel is that is the primer. So the definition of biotremology as a discipline, 2016. So now I enter more in the. I have to check time here. So I guess I have enough time. So Scaphoideus titanus is our model species. It's the American grapevine leafhopper, and is a big problem because it transmits a disease. That is a phytoplasma that killed the grapevines. Yeah, we have another model that is Empoasca vitis, that is similar to the Empoasca fabri that we have in USA. It's a leafhopper, a green leafhopper, and in this case, this causes massive infestations and not any kind of transmission of diseases. So here I have a video, but it's not working, the audio, but I can show you, however, that is an Italian, but it's a car ship duet. You see that the male is here, is vibrating, this is the female, and the male is searching and conducting the duet. So there is a duet, one call, the other respond, and finally the female never moved during the full process that on average is 5–10 minutes, while the male reached for the female and then they mate. So basically this behavior is very interesting. Let me see if it seems that it's stuck. Okay, it's not good. Uh-oh. Can you still hear me? I think I have to. Can you still hear me?
Yeah, I can still hear you.
Okay, I tried to restart the sharing because let me see, because it was stuck. Yeah, but it's not working anymore. There is some issue. I don't know. It was not a good idea to start the video, I guess. Yeah. Try again. Okay, but if I go to the next slide.
We're just seeing the video, Valerio.
I know. I'm trying to move to the next slide because the video is a bit, it's the problem. If we move to the next, we are safe. Now I don't see anything anymore.
I went ahead and stopped your sharing so you could try to share it again.
Yeah. Better not to use videos and these kind of things, I guess.
Yeah, I think we're just seeing the black screen there.
Yeah, it's
Let me stop your sharing again and let you see if you can go back into the screen.
yeah. Try again.
Yep, we're seeing your face at the moment, Valerio.
There is a problem with this. There's a problem here with my computer, I guess. Because here, I'm trying to close at least the PowerPoint.
Yeah, you might just restart it and we'll try to pick up where you left off at.
Yeah, I can try to restart everything, I guess.
Okay, yeah, it sounds like your video may have kind of caused some other things to crash. Yeah, it looks like. So we may have lost Valerio for a little bit. We'll give him a moment or two to get back online with us. Yeah, I think we lost him. Give him a moment to get back online. For those of you waiting for Valerio to return, I'm going to mute my mic for a little bit and we'll wait to see if he logs back on and rejoins us here in the next few minutes.
Okay, I'm back. Can you hear me? Oh, can you hear me? Hello?
Yep, we can hear you, Valerio.
Okay, okay. And now I try again to share and see. Okay, now I put in the presentation
Oh,
mode.
there you are.
Okay, so I'm
Great.
sorry for this. So basically, how works the vibrational duet between a pair of scapuloidales titanus? You can see here a spectrogram and oscillogram, and from this it's not very clear what happens. But if I go in more zoom, you will see that there are pairs of signals. The blue one is the male and the red one is the female, and there is a specific temporal pattern of alternation between males and females. This means that if the female fails to reply within a certain time window, that in this case is 0.20 milliseconds, the male stops searching. He doesn't believe she's a real female. So the species specificity is very high in this species. But this is good news for us, because this means that it's even possible to try to create trouble to them. We started to study this model in 2006, when we started with the mating behavior, of course, and vibrational signals. This is a technology readiness level map. Basically, this means how the technology associated to a device, a new solution, an invention evolves according to the step. Because step 1 is the basic research, while if you go further to step 9 and 10, where you have the full product. Of course, in this moment, 2017 update is more or less the same, 2019, we have commercial orchards that are working with our prototypes. And this is fruit of intense work that lasted more than 10 years and started in the lab and started from basic observations of mating behavior of the species. So what we describe for Scaphrideus that this duet is not the only one. There are basically 3 main duets in the species. If I go here on the right, you see that the basic behavior of this guy, of the male, is to do a mating calling song where this tries to find females in the environment. And this behavior is called call and fly. So if you get a response, you start to do it and go in the next step. Otherwise, it jumps around and jump and jump and jump as long as it doesn't find the female receptive. The next phase is identification. Identification phase is a bit complicated, and you can see in the papers what it's about. But if identification is accomplished, then you go to the location, it's another duet, and finally to the courtship, that is what we've seen in the video. So it's very complicated. And this behavior is kind of mediated by phase-specific vibrational duet, where the temporal pattern, as I showed you in the previous image, is very important. And the duet is maintained from identification to courtship. So only in the copula they stop emitting duets. But this guy here, the N, is very important. What is the N? It's the disturbance noise. Who's making the disturbance noise? The males, rival males. So if it happens that 2 males are on the same leaf and one is in delay in the communication, so it comes after the first male, we try to interrupt them using a specific disturbance noise. And when this happens, the communication is interrupted indefinitely. So this means that it takes some minutes before one of the 2 males, we don't know if the first one or the rival one, they start a new communication. But this means that there is a stop. So that's very important for us. This is just the spectrum of the disturbance noise. And you can see from this image, this is the call and this is the single pulse, and the disturbance noise is covering exactly the same frequency range because it's going to overlap, to mask the signal of the male. This is an example how it works. This on the left is a male and is sending these vibrations through the leaf, and the female responds and they makes a duet. But if there is another male, this starts emitting the disturbance noise and everything is stopped. So this principle is very interesting because if I replace this male with a shaker, I have the same effect. So you understand that this opens a lot of new possibilities for us. Because if I can introduce the disturbance noise in the environment, I don't say I eliminate the mating, but I can significantly reduce it. So what we did was to study the mating behavior, understanding signal function using playback tests, for instance, in the lab. And as a step 3 is a lab test of mating interference, okay? And that's what we did. What happens in the lab if I try this approach, okay? Then we go to the field, step 4 and 5, and we try. You see, this is one of our first prototypes that was hung to the wire, and this is a vineyard, and we just put our insects inside the sleeves to see if they made it or not. We found many important information still in the lab, like a threshold of amplitude or intensity that let the system work. because if we are over this threshold, we have 100% or 90 at least of success in a small environment like a sleeve. And you can imagine that if we work at an open field, this should be also better. This is a photo, a picture. Our sleeves are very small. There are 3-4 shoots with about 5 to 10 leaves, full-grown leaves, and we release the couples, single couples inside, one male and one female. And then the next step is to try to apply this technology to a commercial orchard. So this is our first commercial orchard. That's the first vibration of veneer, and you see it's about the size 1 hectare, 1.4 hectare. And this is organic, good old trellis system, so it's a very modern system, and we put 2 shakers that are the emitter of vibrations, of disturbance noise vibration per row. So as a whole, in this area, we have 110 shakers. And of course, we have control all around. This is our experiment. This is a picture. You see, this first one, this first veneer is wired. So this means that we are taking electricity from a power plant. We are not using a solar panel. Although in the next 2 veneers, because we have now 3 veneers, we are using solar panels. So we are not using electricity from facilities. This is the shaker. In this case, our next evolution was to apply the shaker to the pole and not to the wire, because we found a lot of improvements if we do this. Oh, I have to say that we collaborate with engineers, electronics people. So it's a multidisciplinary team where entomologists work together with engineers, because otherwise we have no chance to do something good. And this is solar panels where we are using in another veneer. And in this moment, we have 3 vineyards in northern Italy, 2 in Trentino, and 1 is in Piedmont, another important wine region. So basically, this is a scheme, and we have 1 pole with a shaker. This is an example of our vineyards. You see in this line that one of these can manage about, in one direction, 30 meters. So altogether, a single shaker can manage 50 to 60 meters, having a threshold of sensitivity that is for us satisfactory. I mean, threshold of, threshold of intensity that is satisfactory and enough to interrupt the mating communication of Scaphoideus titanus and Empoas cavitis. I didn't talk about Empoas cavitis much, but the green leafhopper has very similar behaviors. We describe them in other papers, and basically our signal include the disturbance noise of Scaphoideus titanus and another band that is designed to cover Empoas cavitis. So it's a multi-purpose device, okay? So what happens that from the pole, the signal is transmitted through the wires, and from the wires to the plants. And this means that a trellis like a vineyard trellis is very good for our purpose, because we have a lot of help from the wires that are all around. Okay, we use laser vibrometer to test and to measure the efficacy of our method, of our device. So this is an example. It works in this way. You have a feral population in the field. What you do is just to apply your shaker. This is an old figure. So this green shaker now it is on the pole, and everything is vibrating. And vibrating in a very specific way, is not random vibrations. I want to repeat it, because we need to be selective. I don't want to disrupt other animals. I don't want to disrupt predators. I don't want to disrupt parasitoids. So we have to use specific frequencies to let it work and to disturb as less insect as possible. Of course, it's not possible to be super selective because it's not a signal that is working for only one species, but still we try to be less impactful as possible on the environment. Okay, this is an evolution of different type of shakers, and the time in 10 years we developed many shakers. This is an example of the system, and I want to give you some preliminary results. We're still evaluating a lot of things like the impact of parasitoids and predators, but I want to just give some numbers. And this is an example of data after 3 years of experiments in the first vineyard, Vascaphoideus. Yes, we have to consider that results. Yeah, we have a difference between control and treatment that is significant in 2018 and 19, because I also expect an improvement with the time. And however, we are talking about one vineyard, so it's too early to be happy about that. But for Empoasca, we have a very good result, especially in 2019, when there was a big outbreak in the control, while in the treated area it remained under an economical threshold. We are not treating. There is not insecticide at all. So the thing is that our vineyards are kept in a condition of not insect treatment. And also in this case, you see that the results are very encouraging. And how can I further assess this effect? Because this is a manual sampling. What we did is just taking 24 plants per plot, 20 leaves per plant, and working from May to August. So finally, you have thousands of checked leaves. Is it enough to assess the efficacy of the method? Probably no. We try also to use a sweeping net monitoring in 2019 only, and we have exactly the same results. Treated area has a double population. I'm sorry, this is a mistake. It's the opposite. The treated area is half population of the control. And this is super consistent with the data that we observed with the manual sampling. And then the other method that we used to monitor was the sticky traps. And sticky traps, we put a ton of sticky traps. For each vineyard, about, it was 12 sticky traps per vineyard. What we found that in this case, the treated is correct. We had many more captures in the treated area than in the control. That is totally inconsistent with other observations that we did. And we say, why? What's wrong with this? So basically, we examined the number of males and females. And you can see that there is a gap of 24% in the general number of captures, but the sex rate is very different. In the treated area, 96% of captures are only males, while in the other area is about 85%. And if I consider the sex rate, along the season, we have constantly that the treated area has a very higher number of males. So our hypothesis to explain this is that the males, we of course standardize the number, did some operations, but finally our idea is that males are flying 3 times more than expected, while females are flying the same. And in fact, the population of females corresponds to what we saw in the manual and the sweep net samplings. But the male behavior is totally changing. And that's very good news for us, because we expect that males are stressed. They don't find a female, and they increase a lot their co-confined behavior, and they're restless. But this means probably that they're also causing less damage. In fact, we found, I didn't show this, I'm not showing this data because they are still under elaboration, but I can tell you that 50% of damage reduction in the treated area has been measured. And this is for Scaphoideus, but the same can be said for Empoasca. It's less the difference. It's only 9 to 13. It's not much apparently, but if you see along the season, you have constantly higher males in the sticky traps. And this, in this case, is 2.4. And also females indeed is significant. So apparently also females seems to be more stressed by this environment. Consider that Empoasca is a Typhlochibina, belongs to a different subfamily of Scaphoideus with this Deltocephalina. So it's possible also that there are differences. But what for us is very interesting that we have a different response of males that are stressed with this technique in the treated area in comparison with the control. So that could be a way to assess the method and if the method is working in the future, because we needed this. We didn't know how to do. How can I check if it is working or not? Yeah, just doing sampling and that are super time consuming, or there is another way to measure immediately that is working. And probably the use of sticky traps to measure the sex ratio is a very good tool, but we have to work on it. These are just ideas that are coming out step by step doing this experiment. I think my time is going, correct me if I'm wrong, we got delays. I want just to show you the agrotraps. So the brown stink bug is an animal that uses both pheromones and vibrations. But pheromones are long range cues that they use to attract from the environment other conspecifics. While vibrations are specifically dedicated to mating behavior. So in general, they aggregate in one place and then they use vibrations to mate. These are examples of signals of females. So a female attracts the male. We developed in the course of some years different type of traps, also again with the help of engineers to maximize the efficacy of the transmission and the spread in the environment. And now we have two prototypes that are working. This is the vibro trap number 1 and number 2, and they are catching more than the commercial traps. But in this case, it is a rescue trap that is very common both in USA and Italy, and the sticky traps. They seem to be much better, but we need a second season to try these new traps because in this moment we have very few prototypes. I want to work with 30, 40, 50 prototypes to assess if they're really working, because having a few prototypes, you risk to say, oh, it's working, but then you find that the location is very important. And in the case of a brown stink bug, even a few meters of distance from each other can cause a lot of difference. So the point is that I need a huge number of replications to assess if they're working or not. And putting together vibrations and pheromones should drive them better in the traps. Why? Because the problem is that the classic trap like rescue and sticky, they use only pheromones and they catch a lot. But most of the animals are outside the traps because they're just gathered there, then they do whatever they want. But vibrations should improve the method because they drive the males, at least, exactly inside the trap. And this is for us very important for early detection or detection at low population level. And for instance, countries where this insect has not yet arrived would find very useful this kind of trap, because my hypothesis is that can work at lower density of population. So it can be an early warning system. But again, these are examples. I want just to give quickly, and then I'm open to the question time. other projects that are going on in our lab, in other labs worldwide. So we are studying here mirids in greenhouses, white flies, and there are different projects. Also in Japan are studying white flies indeed, spittlebugs, and sharpshooters in collaboration with USDA California. I had no time, but this is also another very interesting project. While other possibilities are about cerambyciid beetles and bark beetles. There are works in USA, Canada, Japan, and we have collaborations also about psyllids. This is another group very interesting that is worthy to be studied in order to find a possible vibrational approach for the pest control. Yeah, there are many people, many projects that are working at this, and we are super happy to, we are very excited indeed to do this because it's a new discipline, a new approach. We are pioneers, and yeah, of course, there are so many things that are not known, but this is a very good opportunity for new scientists, for young people that want to expand this, to extend the knowledge of this discipline. There is a lot of field. And so I hope that this short presentation, for sure, is insufficient to explain the proper way the subject, will give at least some hint of curiosity in the listeners. So thank you very much for your attention, and I'm ready to respond to questions.
Great, thank you, Valerio. I'm gonna open it up for questions for anyone who has a question for Valerio. If you want to type it in, I can pass that along to him. Valerio, can I ask you a question quickly? We can go back to your screen if you're there.
Okay, I share it again.
You can go back to your camera if you like.
But you want me to share, or what do you want from me exactly?
Just your camera would be fine.
Okay, let me start videoing this. You see if it works.
I think there you are. Yeah.
Yeah.
Yep. So a quick question for you. I'll wait to see if anyone else has questions in the chat box. But in a lot of different interventions using pesticides, sometimes we see some sort of evolutionary response to where they become, for lack of a better word, accommodated to the pesticide. Do you think that's a potential for using noise, that there's some evolution for resistance to noise over time?
I think it's too early to say this because we are doing this approach only from 3 years, and for the moment it's going well. What I can say is that unlike chemicals, where you have molecules that are very specific, we are using signals that cover a rather large range of frequency, not super large, it's a bit more than the insect carrier frequencies. So we try to be large in order to avoid a quick shift from a carrier frequency to another one. This should help in preventing, according to us, an easy resistance induction. But it's really too early to understand if something is going to happen in the long term.
Great, thank you. Let me see if there's anyone else here at UNH with questions. Grant?
And male stink bugs made with multiple females.
Okay, so I have a question from here at UNH. There was a question about male stink bugs having multiple mating opportunities with females. Is that typical?
Yeah, it's typical, but in fact, in this case, we won't just trap them. So I want to catch them. And that's good, because if they mate one time and then they are not available anymore, this means that I can't catch already mated guys. However, our system of trapping is dedicated to males.
So wouldn't that still be like a pretty favorable sexual composition for a population now? To have more females than males, that they can mate multiple.
Oh, I guess the question here was also about if there's more females still in the population, is that going to have an applied impact as much if you're just pulling males out? I guess would be the question, right, Grant? In other words, if you're biasing to capturing males and yet still leaving mostly females, do you expect to see a measurable effect on lowering the impact of the pest in the
no,
field?
Because this is not a mass trapping system. It's for me, it's very important tool of monitoring for taking decisions. So I guess that is much more precise than the currently used commercial traps that are based on pheromones that are not very precise at all.
Okay.
It should be more specific, and so giving information more important for taking decisions.
I've got one more question here. Let me see if I see another one come up in chat here. Okay, so there is a question that came up in chat, and we talked a little bit about measuring efficiency as it relates to the pests, but how about mechanisms for assessing the degree of effect on predators? For example, might this disrupt spider populations? And so, does that matter? How would it be measured? In other words, kind of peripheral unintended effects on other invertebrates in the system.
Well, what is very interesting that we found, because we had a crew of students helping us in the last 2 years, and each group of students was dedicated to a different insect group. So there were guys and girls that were studying the impact on predators, parasitoids, thrips, and generic plant visitors like Hymenoptera or butterflies, whatever. So what we found is that there is not any effect in terms of number, because we were collecting them and checking them also, of course, on the sticky traps. And we didn't find a difference in spiders, and in predators like lacewings, we didn't find a difference. On the other hand, we didn't see any difference also in occasional plant dwellers, but we found a difference in the presence of Thysanoptera, thrips. And what we found is that in the sticky traps, we caught many more thrips in the treated area. That is totally consistent with our data with the leafhoppers caught in the treated area. So we didn't study this yet because we had just these numbers. But the idea is that this method can really compromise communication of thrips. And but however, there is no project in the world about thrips. That would be a very nice field of study to see if it's possible to interfere with the thrips.
Very good. We had one other question about the economics of this. How does the cost of potentially installing an array of shakers compare to more traditional methods, including pheromone traps?
At the moment, it's more expensive because we have just prototypes. But the idea of the company that is financing is to make it comparable with the other systems, like, for instance, irrigating systems or other stations that are present in the field. It's just to install something that is long-lasting. Not every year to go there and put new shakers. You buy the system, you keep it for at least guaranteed 5 years, and then you can really compare the cost with other systems. In this moment, it's difficult to make an evaluation because you work with prototypes. There is not an industrial production, but starting from 2020, we are going to have more vineyards also in France. We're going to make some vineyards in France. That is a very good achievement because France is probably the most important viticulture area in the world. And we are very happy that our method is starting to be believed. I mean, if you understand what I mean.
Yeah.
And so when you will have a mass production of these devices, probably what we expect, because the company, of course, wants to be in the market, is to be comparable with, for instance, an irrigation system. So they can choose to put in. And however, I want to say one thing, that we didn't remove the total populations of the leafhoppers or planthoppers from the field. This can let me think that this method can be integrated. It's not something that is a solution that say, okay, I have these, I have no problems anymore. I think this is integrated pest management concept that must be considered also in this case.
That makes total sense. Valerio, I want to thank you for providing the seminar today. It's very, very interesting, thought-provoking. It's got me thinking about spotted lanternflies and how that might be a integrated approach for that kind of impending threat we face here in North America as well. So we'll be posting for all of you that are logged on, we'll be posting the seminar probably later today or tomorrow that you could pass on. We'll send out a link to all of our participants so you can share those with your students and your colleagues. Valerio, appreciate it, and we'll let you go now, and thanks again for providing a seminar for this month.
Thank you very much for inviting me. I'm very happy to be here with you and to share these things with the students.
Thank you so much, and we'll be talking again soon, I'm sure.
Sure.
All right.
Okay. Goodbye, everybody.
Goodbye.